My name is Wyatt Acebedo, and I'll be showing how I established a protocol for Phytophthora identification for Bellarmine. Every year, something goes around slowly killing out plants, crops, you name it. A common plant microorganism, meet Phytophthora, the silent killer of your local plant life. So first, Phytophthora. We should mention the fact that it is a plant-damaging water mold, the same disease that caused the Great Potato Famine in Ireland in 1845. They can reproduce sexually and asexually. They can spread very easily and cause problems for farmers, such as root rot or branch dieback, where nutrients are blocked from entering a tree. This damage can be very taxing for places such as nurseries. For example, if a nursery becomes infected with a species called Phytophthora ramorum, it could lose up to 90,000. It could lose 90,000 to 1.3 million dollars in costs for crop destruction and cleanup plus loss of sales. Phytophthora can be difficult to identify because it can be easily mistaken for other diseases. Bellarmine Marine Chem projects also previously have involved identification. The latest was a student who went through the whole process of identifying in our lab. Now, Phytophthora is a genus, so this means there are many different species under the Phytophthora genus. This list includes only some of the species of Phytophthora. There are many ways Phytophthora can spread, usually through the movement of water. Once on a plant, it makes its way through into the host. The Phytophthora slices through the tissue, then manipulates an immune response from the plant. In the host cell, then starts the process of endocytosis. A vesicle is formed to drain the plant of its nutrients, which causes the plant to wilt. The overall impact of this plant disease is in its commercial usage of plants, specifically nurseries and farming industries. This image shows the difference between a field treated with fungicide, shown on the right, and untreated on the left. The treatment of fungicides is shown to become less effective, which shows how it's becoming harder to control and manage the disease. The effect of Phytophthora on industries is expected to increase with time. What is being done to get rid of it? There is destroying the plant, which when it is infected, because there is pretty much no coming back after infection, steaming the plants or the diseased areas, which can also kill other things like the plants, which is not good. Management practices such as maintaining clean facilities, avoiding overly wet conditions, clearing out any possibility of Phytophthora spreading. The gap in knowledge here is knowing the specific location of where it is. The skills taught in marine chemistry sophomore, freshman year with microbiology and biotech can be expanded to include more practical uses like Phytophthora identification. My problem statement is that protocol towards identifying Phytophthora wasn't previously made for someone with inexperienced lab work, but is now accessible for students to learn specifically where Phytophthora is in agricultural areas. By making a clear way to identify and locate its range, students can learn how to prepare and identify it. So my goal was to adapt WSU protocol to Bellarmine. Students will learn how to use their skills and develop in biotech. Also, I designed a laboratory space for the use of identification on campus. So here is what everything involved. So I started my lab work with learning and going through the biotech piece, and then I learned how to collect the samples. Then I conferred with one of the instructors at Pierce College to understand the process, because they are also doing similar things with Phytophthora in their classes. Over an extended period during the summer of 2024, I spent my time in the WSU Extension Center learning how to use lab equipment in order to identify Phytophthora. My mentor grew the Phytophthora and provided instruction on what to do with the lab equipment. There are 4 main parts to the project. The first is the sample collection of the leaves. Next is the isolation of Phytophthora. Then the extraction/duplication, and lastly, the identification. For each sample, a location for collection would be determined, and about an inch worth of soil would be gathered with a small shovel. A bottle would be filled with water. 2 healthy rhododendron leaves were placed with the sample and rested for 2 days. And afterwards, symptoms of Phytophthora were isolated onto a media plate for further growth. Afterwards, DNA is extracted with the use of a kit called PrepMan Ultra. And then afterwards, sample quantitation is necessary sometimes if the equipment is available, but this is for seeing the concentration of each DNA sample and if the DNA is able to be viewed, but it is not necessary for completion of the protocol. Polymerase chain reaction, or PCR, is used to make copies of the DNA. For making copies, main components were added with the DNA gene to get a certain strand of the DNA. Primers were then taken from a freezer and used in a flow hood to ensure a sterile environment. Small tubes were labeled and given a corresponding amount of reagents and mixed to create a master mix. These were then placed into the thermocycler to facilitate the DNA amplification. Then there is the electrophoresis piece. Essentially, electrophoresis is the separation process to show the strength of how well that specific part of DNA was amplified. First, a gel is made to hold the DNA, and other mixtures are added in order to see the strengths of amplification. These gels are then run at 130 volts, and their colors and brightnesses are viewed. The final solution is then sent to a sequencing lab. The people at the lab find the DNA sequences and send back the results. Then they can be viewed and analyzed by a program to relate your sequence that you found from the DNA to other existing DNA sequences. In order to do all of these steps, however, it was necessary to create a lab station and take inventory of equipment. Here are some of the tools that students would need to identify Phytophthora. Also, a proposed lab setup was made for the use of culture sequencing. Here is a list of costs I put together for making the lab more accurate, but technically it can be all done on campus. The main objective of this project was to find a clear way of identifying and locating the ranges of Phytophthora in the communities, as well as proposing a protocol with a lab station setup. Tests were done in order to determine the intricacies of the identification process. By going through the process of the protocol provided by WSU, I was able to create an 8-page step-by-step protocol for future Bellarmine marine chemistry students, including overview, tips, safety reminders, objectives, a list of student materials, and pictures of the process. They can contribute to the well-being of the earth by looking for Phytophthora. The original WCU protocol was made for the use of a WCM employee, so it was necessary to make it applicable for a student, for them to apply learned skills from marine chem. Further improvement of this protocol is possible in further studies, as well as further researching the impact of Phytophthora in communities. The building of lab technique begins for marine chem students around sophomore year with sterile technique, pipetting, and other lab skills. This lab should take around 4 to 6 weeks, with waiting for Phytophthora to grow taking most of the time, and actual lab work taking only about 4 days. But planning the lab is really based around the scheduling of marine chem. Also, Pierce College is doing pretty much the same work in some of their classes, so this would prepare students for college as well. Some of the limitations that have made it a little harder to establish the protocol are the equipment limitations in the lab, scheduling meetings at other facilities to work at. Also, the protocol has not been put into practice yet at Bellarmine and has not been used in a class setting. Also, the process of learning the protocol did take a long time. Further research on this topic includes the protocol being utilized by marine chem students. Students could potentially gather data about areas and their Phytophthora statistics. For example, practicing different collection techniques, sampling different areas in Tacoma, using different sampling techniques of Phytophthora like water baiting. So there is potential for further research in methods of collection. Thank you to everyone that has supported me with this project. Thank you to my family, my mom, my dad, and my brother, my friends, my mentors, Dr. Elliott, Ms. Taylor McNeese, Mrs. Lana Hanford, and Ms. Daphne Mary. Thank you to everyone that showed up to support me today. And thank you to Mr. DeGrow for really helping me through this project. Thank you to the class of 25, Marine Chem. I'm so glad I've got the opportunity to know all of you and to travel to Hawaii with you guys. And here are my citations. Questions?
Ask some really hard questions, you guys. This phenomenon is totally waterborne, is it airborne,
In this area specifically? Or just anywhere?
I guess first point is it waterborne is through the water system, or is it airborne as well? How does it move through planet Earth?
So the question was, how does Phytophthora move throughout Earth? And I think it's mostly through water, traveling through different areas, but I'm pretty sure it could be spread through air, but I know it's mostly water-based. Yes?
When you got the DNA sequence results, was it a match to the documented sequence for the species? And were you missing any DNA strands when you
Are you asking if the DNA matched the species? I actually didn't do that part of the protocol, but I did the biotech piece just leading up to that part. I didn't get to the sequencing part, but yeah, I just kind of going through the understanding of how to get to that part. Yeah. Brad?
Why did the learning process take a long time?
The question was, why did the learning process take a long time? So it was a little difficult trying to get scheduled meetings with different mentors, but it just took a long time in the lab trying to read through the protocol and do what I'm being told to do. But yeah, because the protocol provided by WSU was not very descriptive. It wasn't made towards a student like me, so I kind of had to go through that process of talking to my mentor. Yeah.
I saw that you had a thing in there that climate change affects it. How does climate change make this worse?
So the question was, how does climate change make this worse? And to that, I'm not completely sure, but I just know that, yeah, with the increase of climate change, it will start. Actually, I think it's because it thrives in hotter conditions, but yeah, I'm not completely sure about that one. Sorry. Yeah. One more question? For me to do the lab work? So the question was, how much did it cost for me to do the lab? And I didn't buy any equipment. I went to the extension center for WSU, so they provided me with the samples, the tools, so it didn't cost me anything, but yeah, we just used their equipment pretty much.